Yeah, sweet. Thanks for having me today. I'm pretty excited to talk about some Meadow Creek work and research that I've been working on for about the last 2 years. Um, and yeah, I didn't put them on this slide because it was getting a little chaotic, but Casey Justice at CRITFC and Becky Flitcroft at the U.S. Forest Service have been sort of helping me through this research. So start off, just wanted to give everyone an overview um, of the Meadow Creek watershed. I should go one slide forward. Yeah. Um, okay, of the Meadow Creek watershed, just so we're on the same page. Here's a map of the watershed there on the left, with the confluence of the Grand Ronde River being on the far right side of that. Oh, cool, thank you. The watershed itself is about 313 kilometers squared, about 73% managed by the Forest Service, mean annual discharge of 3.65 cubic meters per second. I just wanted to point out like a couple important creeks. So we'll start at the headwaters. There's Walkup Creek that comes in as the first named creek to the Meadow Creek tributary. That brown dashed outline is the Starkey Experimental Forestry Range. Then as we head downstream from there, there's Bear Creek, Battle Creek, Burnt Corral Creek. And then we get to the confluence with McCoy Creek. That's McCoy Meadows, and I'll refer to that a couple times throughout the course of this talk. And that is owned and managed by the CTUR, or yeah, Confederated Tribes of the Umatilla Indian Reservation. Only steelhead currently spawn within the watershed, and then there's sort of a long history of degradation dating back to the 1800s. And I'll actually walk through that now just so we're on the same page, do a little bit of a history lesson. So 1820s to 1830s, there was extensive fur trapping in this region, Grand Ronde watershed as a whole, but for sure in Meadow Creek itself. And then in the 1850s is really when the first homesteaders came to the Grand Ronde watershed. By the 1860s, there's a road up to the Meadow Creek. So, access is then increased. There's more people traveling in and through and around. 1870s, we have the settling of Starkey, the town site. And then in the 1880s, McCoy Meadows is actually homesteaded. Throughout this sort of time period, 1870s to 1900s, there's really heavy grazing by free-range cattle and sheep, and then actually cattle drives being driven from as far away as Idaho and Wyoming up and over the Blue Mountains to get to the railroad. I put these photos in here on the right just because I thought they were really pertinent. So at the top is 1902 and the bottom is 1997, and that's Fly Creek Meadow. And you can see in 1902 already there's been pretty denuded riparian vegetation. There's like bare ground, like pretty impacted conditions already um, in 1902. Oops. So continuing on a little farther, we kind of have the start of logging within the basin. In the 1890s, there's splash damming of Meadow Creek and Dark Canyon Creek. That's a photo of a splash dam on the Minam, just to give you some like scalar context. And then in the 1920s, there's railroad grades being built up various tributaries of Meadow Creek and Meadow Creek main stem itself. Then in the 50s, truck logging starts. So we have the construction of roads, and there's over 30 square miles of Meadow Creek that's owned by the U.S. Forest Service that gets logged. And then in the 60s to the 80s, there's a pretty major change to the lower part to McCoy Meadows, where it's ditched and straightened in response to some pretty severe floods in the late 50s into the 60s. So with all that sort of history of degradation set up, we can actually look at some sort of statistical comparisons that were analyzed or conducted in the Meadow Creek watershed. So there's a 20% loss in large pools in Meadow Creek itself, 50% total loss in pool habitat. Those numbers are much higher in McCoy Creek with an 82% loss in pools and 73% loss in pool habitat. And then another study saw a pretty large increase in stream widths, anywhere from 1.23 to 1.85 times increase in stream widths, just really pointing to a loss of complexity within the system. These are some before and after photos, not quite of the full straightening alignment in McCoy Meadows, but of it around that time. Within the context of this degradation, there's diminishing summer steelhead returns. So here on the y-axis, we have Meadow and McCoy Creek's combined index red count being conducted by ODFW on a yearly basis. And then that blue trend line showing that, as we're going through time, we're actually decreasing the number of reds being counted within the basin. That red star is 1997 when Snake River summer steelhead are listed as a threatened species. So, based on all that context and declining steelhead returns, there becomes interest or efforts to begin restoring Meadow Creek in the late 1980s. Projects are developed, thought up, put into implementation. So, in 1990, the first sort of large-scale restoration project occurs within the Starkey Experimental Forest and Range. That's targeting about 3.7 kilometers of Meadow Creek main stem that goes through Starkey. In 1997 to 2010, a large project is really designed and implemented to restore the original character of the McCoy Meadows confluence. So that's removing the ditch or unditching the stream, remeandering the stream, installation of extensive large wood. Some of those boulder weirs are installed, a new bridge is installed over the creek. Really just sort of a widespread, multifaceted approach to try and restore that meadow system. Continuing on to more present or more modern era, from 2012 to 2014, there was some extensive retreatment of Meadow Creek within the Starkey Experimental Forest and Range. That constituted about 7.2 kilometers. They got retreated with large wood additions. There's extensive riparian planting along that corridor. And there is the construction or reconstruction of 9 separate exclosures, which we'll talk about a little bit later. But just for a rundown, there's essentially 3 grazing treatments: snow grazing, cattle only, and then deer and elk only. So with that sort of background set up, these are sort of the goals of my talk. Talk about 3 different essentially data sets that we're looking at the efficacy of these various restoration actions. So the first one is looking at Oncorhynchus micus outmigration dynamics following that early 1990s restoration project. The second one is looking at O. micus habitat in juvenile abundance after the mid 2010s restoration. And the third, again, looking at that mid 2010s restoration, but trying to get at riparian vegetation response and see how things changed after those projects were implemented. So, for objective number one, most of these outmigrant traps were actually these Humphrey style, like not rotary, but inclined plane traps. So I thought it was cool to throw a photo up there just so everyone was on the same page. So trapping was conducted by the U.S. Forest Service for a number of years, from 1987 to 1999. The pre-period would be 87 to 90, with the post-period being 1991 to 1999. There's sort of a variable number of traps depending on the year in question, and I've labeled the traps on the map here based on their stream kilometer. So 0 is just upstream from the confluence with the Grand Ronde. And then we have KM5, which is just upstream of McCoy Meadows. So that's just Meadow Creek, not including McCoy or Dark Canyon creeks. 25 is just inside the Starkey boundary. And then 33 is just below the narrows of Meadow Creek. KM0 and KM5 had the longest trapping histories, so I'll talk mostly about them. And then in addition to your standard biometric data, mark recapture approaches were used to essentially estimate trap efficiency and outmigrant numbers. This little purple circle, I'll use that a couple times in this talk, and that's just me highlighting where restoration actions took place. So that's approximately the 3.7 kilometers that were under restoration. So just to take a look at some biometrics first, on this plot, we have Fulton's condition factor on the y-axis with x-axis being years, and that dotted line will be the approximate time that restoration took place. We can see that there's not really a huge change after the restoration, although there is a slight increase from an average of 0.96 to 1.02. These blue numbers just indicate the sample size for all of these data. Similar story going on with fork length, although it is a little more, I would guess I say ramped, where you see right after the restoration an increase in fork length that then kind of drops back down to what was about the average pre-restoration. And then that same story is going on if we look at fish weights. Yeah, so pretty consistent story between these three different metrics. If we look at the actual efficiency of the trap, so this is on the y-axis, we have trap efficiency, and then on the x-axis, we have the year. This dotted line going vertically is the restoration, when the restoration occurred, and then I've highlighted in red 5% trap efficiency. Traps that are lighter in color are farther downstream, with traps higher upstream being darker in color. What you can essentially see here is that traps farther downstream were more efficient. And then the pre-restoration period, trap efficiency was really poor. I use this 5% cutoff essentially because um, if we got lower than 5% trap efficiency, our confident intervals were getting very large. And then we weren't confident that we were meeting the assumptions of mark recapture at that point. So these are actually the estimates produced from the trap. You'll notice that the time period is constrained, and that's because if traps were less than 5%, they weren't included here. So we have the total number of Omicus outmigrants on the y-axis, with traps again colored by their position in the watershed. This time we have darker colors being more downstream, lighter colors being more upstream, and then the confidence intervals are actually shaded in the next to the lines out there. It's really hard to assess whether there was a effect given our lack of data before the restoration actions, but there's no clear night and day difference here. To dive into this a little bit more, I took a look at just the lower most downstream traps where we had longer data periods. And what we could see there is that there was a difference in the average. However, the sample size between those two periods was quite different. And then the confidence intervals obviously were quite large. It's difficult to say for sure, but I'm thinking that there was little to no impact on actual outmigrant numbers. And then finally, because of the sort of sequential nature of these traps, we can use them to investigate untrapped or unmonitored areas. So a good example and one that I'll dive into here is with trap 0 and trap 5. If we know how many fish are out migrating at trap 0 and we know how many fish are out migrating at trap 5, we can subtract the fish from trap 5 and get an idea of what is going on in McCoy and Dark Canyon creeks that weren't monitored. And so that's what this plot represents. On the y-axis, on the top plot, we have the number of Omicus outmigrating, and that's color-coded by Meadow Creek fish, i.e. the ones from Trap 5 in purple, and then the other fish, i.e. ones that we couldn't account for by Trap 5, presumably coming from McCoy and Dark Canyon creeks in yellow. That same sort of color gradation where the darker one is Meadow and the lighter one is McCoy in Dark Canyon is down below, but broken out into percent. And what you can see is there's pretty high variation from year to year in terms of what is the dominant exporter of fish from the watershed, i.e. which tributary is being more productive. And so just some stats from that. Meadow Creek's estimate varied by a factor of about 6, Meadow Creek being that trap 5 above McCoy Meadows. McCoy and Dark Canyon estimate varied by a factor of 50, while the total watershed factor only varied by about 5. So I took that to mean that there are some portfolio effects going on here, whereby the total watershed outmigration number is more stable than any of its individual parts. So just to kind of wrap this section up and take some conclusions, we saw some small increases in Fulton's condition, length, and weight, but they weren't long running. There's no clear increase in outmigrant numbers, and then there are some portfolio effects going on with the individual tributaries to watershed stability. So jumping into my second objective, really wanted to figure out if the more recent restoration actions had caused any substantial changes to Omicus habitat or abundance following the large wood restoration. And I limited this to 2011 to 2017, reason being is we had champ surveys throughout that period. Large wood and riparian planting was installed 2012 to 2014 within the Starkey Experimental Forest and Range. Again, that's outlined in the brown sort of hatching. and then the purple being the actual reach that experienced restoration work. We had CHAMP study sites, so those within Starkey we treated as controls. Within Starkey we're treated as treatments, and outside of Starkey we're treated as controls, with before and after periods set individually per site. But it worked out to about 2011 to 2013 is before, 2014 to 2017 is after. And if you aren't familiar with CHAMP, they collect a variety of fish habitat metrics, topobathymetric data, and snorkel count data. And we further refine that snorkel count data using a Bayesian detection model that used habitat covariates to account for imperfect detection while snorkeling that a colleague at CRITFC, Ben Stanton, developed. And then this box down here is just our approximate sample sizes. And you can see how many times each site was surveyed based on the color coding. So how do we analyze this data? Mainly through ANOVA analysis, where we could look at period and treatment and then their interaction between each other. Um, we log transformed the variables where necessary. And for juvenile Omicus, we standardized the fish density. So we divided by the meters squared of available wetted habitat, and then divided that number by the number of reds observed in ODFW's index counts the year prior. We also examined this data in a negative binomial GLM to try and get a better idea of what was driving these um, sort of these abundances within particular sites, to try and get a better idea of whether certain habitat variables were important or it was just sort of intrinsic production. So what were our results? So for the habitat assessment, we found many differences between treatments. So the control and treatments, there was a significant effect there, but not in period, and then not in the interaction between the two of them, indicating that maybe there are some systemic differences between our treatment and control sites. There's only two significant interactions between period and treatment, which would indicate an effect around the time that the restoration took place. And I will run through those real quick. The first was substrate D16, where we saw some finding at the control sites, and the impact sites remained pretty much the same. Additionally, we found the total fish cover increased at impact sites slightly, while it decreased slightly at control sites. If we take a look at the juvenile data, this is sort of a violin plot where you can see the distribution of all the data, with the blue being the mean and plus or minus the standard error. What you can see is there's the control pretty much doesn't change between the before and after periods. So left is the before, right is the after. The control pretty much doesn't change, while we see a slight increase in the impact. But if you look at the y-axis, which is Omicus density divided by the red count, these are like very, very small changes. So this came out to be not significant within a ANOVA analysis. There was a significant difference between the treatment types, but no difference between periods and no significant interaction. And then I'll just point out that there's outliers in every single one of these categories. So that likely impacts the result a bit as well. So now sort of into the meat of this analysis, I conducted a generalized linear model analysis where we were trying to figure out what the best performing predictors for Omicus in the system would be. So we kept the Backy effects being period, before or after, and then treatment, impact, or control. And the best performing model also included an index of steelhead reproductive effort. So that was the number of reds that were produced within the system the year before, and the number of wild steelhead that were passing Lower Granite Dam, mean annual flow, and then maximum weekly maximum water temperatures. And if we look at this data within a marginal effects context, so here when I'm talking about marginal effects, I mean we're keeping the mean value of all the other variables in the model, and then just allowing the variable of interest to vary by one in any direction to get an idea of what that single variable, to get an idea of the impact of that single variable. We see that mean annual flow, an increase of about 1 cubic meter per second would result in about 1.05 times increase in Omicus, whereas a 1C increase in maximum weekly maximum water temperature would actually decrease Omicus by 0.68 times. Then if we look at that steelhead reproductive effort index that I produced, it's pretty impactful. So like an increase of 1, which would be very large because it's Z-score transformed, translates into about 3.78 times increase in Omicus within a site. So this is sort of an average site. And then if we just look at it within the context of the Backy design, we actually find some pretty interesting and unexpected, at least for me, results that the controls don't really change between the before and after. There's a difference of 3 fish, whereas there's actually a decrease in the impact by about 20 fish. The confidence intervals overlap pretty heavily. That's not super. It's not significant, but an interesting finding nonetheless. So takeaways from this section is essentially that the restoration caused little detectable change to fish habitat. There was some small increases in fish densities. And I think really most importantly here is these intrinsics or sort of like long time horizon processes are the best predictors of juvenile omega densities. And what I mean by that is like mean annual flow is something that's very difficult to manipulate in a system that doesn't really have any dam impacts, or it's really hard to change your maximum weekly maximum water temperature. And if you could, it would take like many decades to achieve that. The habitat variables that are more easily influenced by restoration, be that fish cover or total amount of wood in the system, were not found to be good predictors of fish abundance. Okay, so on to section 3. So this section, I wanted to take a little sort of look outside the stream and look at how riparian vegetation changed between 2009 and 2020. And the reason for those two dates are LiDAR flights. I have an error on this side. It says 2011, but it's 2020. So 11 years, 2009 and 2020. The way that we calculated riparian vegetation data was mainly focused on vegetation height. So we took the highest hit from LiDAR data, subtracted the bare earth returns, which gave us vegetation height, and produced data like you can see on the left here, where you can see individual trees and then sort of preponderances of bushes, etc. I wanted to assess how growth varied across different valley bottom segments, restoration treatments, and exclosures. And so to do that, I produced a total of 1,186 transects that ran perpendicular to the Meadow Creek Valley bottom from the mouth with the Grand Ronde all the way up to above Walkup Creek, only continuing on Meadow Creek main stem itself. These were then what's the word here? These were then clipped to the valley bottom floor itself so that we weren't measuring trees that were on the hill slope or the transition zone between the riparian area and the hill slope itself. Okay, so this is a bit of a confusing figure. I should have made a better one, but I'll walk us through it so we're all on the same page. I used some valley bottom segments that were delineated based on geomorphic and land use characteristics for restoration purposes, but they fit pretty well, I thought, within the context of work that had already occurred and then sort of the general scope of Meadow Creek itself. So as you move downstream from left to right, the numbers increase, indicating just larger basin area essentially. So Valley Bottom 2 starts above the confluence with Walkup Creek and then goes to the start of the Narrows. Valley Bottom 3 is the full section of the Narrows of Meadow Creek. 4A goes from the Narrows to Bali Canyon. 4B goes from Bali Canyon down to, 4A2 goes from Bali Canyon down to the Starkey Experimental Forest boundary. And then we have 4B starts from the boundary down to just above Battle Creek. 4B2 from Battle Creek to Burnt Corral. 4B3 from Burnt Corral to the start of McCoy Meadows. All of McCoy Meadows is in Valley Bottom 5, and then everything below McCoy Meadows is in Valley Bottom 6. And yeah, this just allowed us to compare different restoration histories and trajectories using this LiDAR data set. So what were the results? I'll run you through this graph first. So on the y-axis, we have change in vegetation height between those two LiDAR surveys. And on the x-axis, we have valley bottom kilometers. So these are slightly different than stream kilometers because the valley bottom line is straighter. 0 being the confluence with the Ronde River, and 28 being above the Aquatic Creek confluence. And then each of these dashed lines represents the segmentation into the different valley bottom segments. Green indicates positive growth, while purple indicates negative growth or lack of growth, I guess. Mean change overall for the data set was positive 1.38 meters. 96% of the transects saw increases, while 59% of the transects saw increases by 1 meter. And then 9% saw an increase by 3 meters, with only 2% seeing an increase of 4 meters or more. If we look at this data in another way, looking at box plots by the individual valley bottom segments. So once again, we have most upstream on the right being valley bottom segment 2, most downstream being the segment that goes from McCoy Meadows all the way to the confluence on the left, being valley bottom segment 6. We can see that there's sort of generally a trend whereby the upper segments are growing more quickly than the lower segments, and that's particularly apparent if we compare VB2 to VB5, which are both fairly wide, unconstrained meadow systems. Um, Within VB2 in particular, there's 2.15 meters of mean growth within that time period. It contained 50% of the transects where 3 or more meters of growth were noted, and 68% of the transects where 4 meters or more of growth were noted. Yeah, as I stated before, VB5 was growing the slowest at just 0.68 meters of growth over 11 years. So there was one more part of this analysis where we had this series of riparian exclosures. There were 9 exclosures within Starkey Experimental Forest in range of 3 types. There was no grazing, deer and elk only, i.e. only grazed by deer and elk, and cattle grazing only. They were about 3 acres in size, and there were 3 replicates of each, and they were distributed through different valley bottom segments. So you can see here that there was 2 pretty high up in segment 2. There was 3 pretty high up in segment 2, 3 pretty low near the Starkey Experimental Forest boundary in segment 4A2, and then 3 and 4A1. In addition to that, CTUIR constructed 26 exclosures in McCoy Meadows. They were established approximately 2009 to 2011, and they were much smaller, about an acre in size on average, but there was no grazing at all in any of those. So because the nature of the transects being incremental, and there was not each exclosure had the same number of transects, and there were much smaller areas, we actually calculated pixel to pixel change detection within the exclosures rather than using the transect lines themselves. So this just meant subtracting the 2020 pixel value from the 2009 pixel value. So here I found some pretty surprising results. The deer and elk exclosures were actually, i.e. the exclosures that allowed deer and elk grazing, but not cattle grazing, were growing more quickly than those grazed by livestock or not grazed at all. That was pretty surprising, and the differences weren't they weren't super large, but the standard errors were pretty small due to the sample size, so they weren't overlapping. Um, if we look at the McCoy Meadows exclosures, they were actually growing slower than any of the treatments within Starkey at about 0.82 meters versus all of the Starkey exclosures were growing at a rate of 1 meter or more over the 11 years. So these exposure results really, they fit well into the sort of overarching valley bottom results that show very similar patterns, whereby the areas lower in the watershed were growing more slowly than areas higher up in the watershed. Okay, so just to jump on some conclusions of this, there's some substantial growth really that occurred across the study area over 11 years, but there was that longitudinal pattern of growth whereby headwaters were going more quickly to the areas lower down in the system, and the exclosures themselves produced some unexpected results where total exclosure didn't provide the most growth, which would sort of be the intuitive way you would think about that happening. It could be that beaver or under other herbivores that aren't actually affected by the exclosures themselves impacted those results by preferentially foraging in those areas or something to that effect. I don't have a full reason why that outcome is, but I'll discuss potential impacts or potential reasoning here in a little bit. So I just want to move to some sort of overall conclusions or bring back sort of my overall thoughts on each of these three pieces. What I found was that outmigrant numbers did not increase notably after the 90s restoration. Habitat conditions were largely unchanged, with minute increases in juvenile abundance after the mid 2010s restoration, and that growth was slower at the bottom of the watershed than it was at the top. So, with that in mind, I want to move to some discussion and I don't know if management takeaways is the best word here, but management thoughts. Um, what is apparent to me is that Meadow Creek sort of lost its wet meadow nature, being a system that was heavily degraded fairly long ago. It experienced a long history of degradation, and so far, restoration efforts haven't been able to restore that. I found this really good photo that actually could sort of document what that degradation looks like. And this comes from Elk Creek in Yellowstone National Park. But you can see on the far left, there's a photo of a pretty wet meadow dominated by willows. There's a beaver dam. There's a bunch of aspen in 1923. About 30 years later, we see a pretty substantial collapse in like the willow community and a transition to a more grassland community. And then 50 years after that, willows are gone, aspen can't be seen, and we have a very narrow sort of riparian area predominantly dominated by grasses, potentially with some sedges and maybe some very small willows. But I think this is a transition that was probably pretty prevalent throughout the interior West, throughout the more arid West, and that documentation on is generally lacking. And something that many areas have tried to fix, have tried to restore. One good example of sort of reversing this process is actually from Whitechoose Creek. And so this is a series of photographs from Whitechoose um, showing from a pre-restoration state to after the implementation of stage zero restoration and the changes in both channel form, but I think importantly, riparian vegetation that occurred in a relatively short time. And so that really gets me to the title of my talk, Stuck in a Rut. And I think it's sort of part of the problem that hasn't been able to be fully addressed in Meadow Creek is both the literal and what I'll call metaphorical ruts that Meadow has been stuck in that the restoration actions haven't been able to lift it out of. So on the left is the stream evolution model, first produced by Clure and Thorne. You might be familiar with it, you might not be. Really, I think the point here is that streams can change and they can go through different stages. And in those different stages, you would expect different channel forms. Um, Within a meadow, you would expect something to be higher up in this chart, i.e. aggradational. And so you're looking at something like a stage 0 or a stage 8 or a stage 1. However, much of Meadow Creek within its meadow areas are as something like off to the right in these segmented boxes. That's a stage 3 or stage 3A, whereby it can't actually access its floodplain at base flows particularly, but even at like 1 to 4 year recurrence intervals. And then the second rut, what I've called sort of a metaphorical rut, I've been thinking about um, ecological stable states and how meadow communities can shift over time. And so what this diagram shows is a classic sort of stable state transition where in box A, you have a community of a certain type, a disturbance happens, which changes that community from one type to another, represented by that ball rolling over the hump. And then hysteresis keeps that community from changing back into its original state without some big outside influence or disturbance. And I think the way that that is sort of manifested within Meadow Creek is limited riparian canopy with a dominant sort of upland species component, and then non-palatable species such as hawthorn or I'm forgetting the name of the other one. Anyway, hawthorn and other species that are non-palatable to grazers being more dominant. And so taking this into like, can future stage zero help escape these ruts? Um, I was trying to think like, what are some examples and can we see this in the literature? This is actually a photo that I took at Camas Creek earlier this spring at one of the BDA projects where it was accessing its floodplain pretty well. However, Meadow Creek was nowhere near its floodplain at the exact same day. So to do this, I took a look at some recent modeling work out of Whiteshoes Creek. So at the top, we have a pre-restoration wetted area, and then at the bottom, we have that same reach post-restoration, and we can see there's way more wetted area, a 300% increase in inundation area during the summer base flow, 450% increase during winter freshets. And then how does that affect fish production? I'm actually skipping this slide just for time. I'll go to the next one. That how does that increase outmigrant production? So they use this same model to look at the number of outmigrant Chinook in various years in the pre-restoration form and the post-restoration form. And what they found was 100-fold increase in Chinook outmigrants on average post-restoration. And then if you look at individual years, some years had no predicted outmigrants in the pre-restoration period, whereas in the post-restoration period, it was fairly consistent. I'll note that the y-axis here is non-linear, so these differences are under-exaggerated. And then finally, getting back to that sort of metaphorical rut, transforming dry meadows into wet meadows, as we saw at White Shoes, there was some really interesting literature that I was reading that came out of a lot of work from Hobbs and Cooper in Yellowstone that was looking at sort of these different ecological stable states. They were calling them beaver wetlands versus elk grasslands, but the way I was almost conceptualizing them was like a fish food stable state versus an ungulate food stable state. And what they found was that browsing intensity actually caused a system transition whereby as elk density increased, they started competing with beaver for food, diminished the amount of food for beaver, leading to abandonment of beaver, collapse of beaver colonies, led to channel incision, which changed that riparian community from one dominated by woody riparian species to one more dominated by lower growing grasses, sedges, etc. An important part of this sort of dynamic was that they also found that groundwater availability in conjunction with browsing pressure was really important to determining whether willows could escape the elevation or the height needed to be unaffected, or I guess less affected by browsing pressure. So here on the x-axis, we have groundwater availability with willow height on the y-axis, and you can see at low to moderate groundwater availability, you're most likely to be in the short willow state, whereas if you go to the high willow state, that's always generally occurring at moderate to high. And then this sort of transition point in the middle, you can be at either, but you're mostly what determines whether you're in either one is the level of grazing or sort of the level of browsing pressure that is being implemented. So overall, bringing that sort of back to the Meadow Creek context, I think that past restoration actions have been there's been a lot of work and effort and well thought out practices done at Meadow Creek, but they failed to fully address the ruts that Meadow Creek was in. And I think part of that was just the long history of degradation and sort of the original starting state that Meadow was in prior to restoration work was very degraded. And I've sort of matched my little logos to what I think was most important for any one particular aspect of my study. But in truth, I think you can't really separate these two things from each other. The stable state of the riparian system is dependent on the position in the stream evolution model and vice versa. So with that, I'll take any questions.
I'll just share. Um, is you Oh, that's right. Thanks for that, Marshall. That's a very thorough review of everything going on in Meadow Creek. Regarding your evaluation of growth within exclosures, did you look at any sort of interaction between exclosure size and growth across that period?
No, I didn't specifically look at size. That's a good question. And I'm sure that there should be an effect there just given how plants establish, and the space that they need to establish in competition among plants and such. But no, I didn't look at that. That's a interesting thought, particularly, I guess, given the difference in sizes and scope between some of the McCoy Meadow ones and the Starkey ones.
Cool, thanks. And if it's okay, I have the microphone. I'll ask him a second question. Um, why use the transect method for the vegetation or evaluating the vegetation response and then the pixel to pixel evaluation for the exclosures?
Yeah. So the transect method was developed because, well, for a number of reasons. First, the computationally, it would have been pretty intense to calculate those over. I mean, it was more than 28 valley kilometers. So we're talking about like a lot of area at a 1 meter pixel resolution. So I think for me it was computationally that seems pretty difficult. And then there is some question of data compatibility, especially when you're looking at LiDAR flights flown like 11 years apart from one another, and trying to get those rasters to match up perfectly is fairly challenging. So using the with the smaller exposures, that's easier accomplished versus trying to straight those across. And the transect method seemed like it would be easy to compare that data to more ground-based data. And like, for example, if I wanted to compare my data to a place where transects had been collected or they didn't have LiDAR data. So it seemed appropriate to use different methods.
Technology, it's complicated, but wasn't expecting that on the mic. Great talk. A question about the smolt trapping data. I mean, that's in essence and kind of like an IMW. What proportion of the anadromous, those were steelhead, right? The data, what proportion of the watershed has been restored? I know that you had 7 miles of this and some other factors. Do you have a feel for the total numbers of miles of steelhead habitat and what proportion has been restored?
Yeah, that's a great question. There's actually a CRITFC staff member who's been working to sort of synthesize all the restoration projects that have been implemented in various tributaries of Meadow Creek over the years. And it is quite complex. I don't have a, like, off the top of my head, first cut answer. But for example, the 4.2 or 3.7, I forget the exact number, proportion that was restored in 1990, that would have been very small. That would have been like under probably 5% of the steelhead distribution. So, relatively small project.
Yeah, the reason I ask is because the studies that have looked at sort of a watershed response, small response to restoration, have basically showed that if you don't restore about 25% or more of the habitat, you probably can't detect a change in the smolt numbers. The other thing is that there's been a fair number of IMWs. A few of them have been very successful, and those really restored almost all the habitat. And so when I looked at your habitat response numbers, you didn't see much of a habitat response, so probably wouldn't expect a steelhead response to that. So I mean, it may be that the restoration isn't working and it hasn't addressed, some of the incision and stuff, but it may also be that there just wasn't enough habitat restored, uh, and there wasn't a big enough change in the habitat to affect the fish response, either at a reach scale or a watershed scale.
Yeah. Yeah. And I certainly agree with you there. I mean, that's sort of where I was trying to go with my, management takeaways is small changes don't equal full restoration.
Great. All right, our last question.
It appeared to be very high variability in adult spawner abundances. And I'm wondering if you did any analysis to look at smolts per red or smolts per spawner in contrast to just abundance. And I'll slip in a couple other questions real quick. In your vegetative analysis, did you look at species composition and vegetative density, or did you control in any way in your growth based on species that were growing?
Okay, so I'll answer the first one. Sorry, the second one first. No, I didn't look at species. I was purely looking at growths, irrespective of species. Didn't control for that. Yeah, I think it would be good to. I just didn't have the data to assign individual species or even really classes of species. And then, okay, so yeah, there's a lot of variability in that adult return red data. And so one of the ways that I tried to sort of circumvent that was to use returns at Bonneville or at Lower Granite Dam as an add-in. The reason being the index surveys at Meadow and McCoy Creek are one day, 2 day index surveys, and so they aren't what you would do to create like a really statistically robust steelhead survey where you would be going out there like every 3 days. And so there's definitely some variability in that data, but I mean, I think the sort of long-term trends hold up. And still within a modeled context, they were more important than, for example, the number of pieces of wood or the stream width or something like that, more traditional habitat variable.
Thank you.